Okay, making sure everyone's all here. All right. Well, it's the top of the hour. I'm Marna Peterson, and I'm your host for the webinar today, titled Current Trends in Antimicrobial Stewardship Through Diagnostic Innovations. This webinar is produced by the Center for Infectious Disease Research and Policy, and we are also supported by an educational grant from bioMérieux. We are super thrilled to have there are two speakers here today, Dr. Romney Humphries. Many of you probably already know her and her work. She's currently the director of Division of Laboratory Medicine at Vanderbilt University Medical Center, an expert in the field of antimicrobials, antibiotic resistance, and diagnostics, both from an industry perspective and an academic perspective across her career. And we also have a colleague of hers from Vanderbilt University Medical Center, Dr. Ritu Banerjee. She is the medical director of the Pediatric Antimicrobial Stewardship Program. They've listed their directorships. They have many other roles and much impact, also serving in societies, as well as being consultants to regulatory bodies. So very thrilled to have them as experts here today. And I'm going to just go through a couple slides, but I think you should be aware that maybe you came to listen just to these speakers speak and take in their knowledge, but you can also go and collect continuing education credit post listening to this webinar. And we're offering credit for pharmacists, physicians, and nurses through our collaborator in our CE provider, MAD-ID. So please check out, go to their site to collect your CE post webinar. So there's some disclosures, and this full disclosure statement for all speakers, I'm not going to necessarily read off the list, but they are available online. And I know Dr. Humphries and Banerjee have theirs listed when they begin their presentation. And as I mentioned, we were very gracious to receive an educational grant from the bioMérieux group to provide this webinar to you. Continuing education, as I mentioned, it is time sensitive though. So after you watch this webinar, you need to go claim your credits within 30 days post watching the webinar. You will go to the MAD-ID site, and then they will send you the credits for pharmacy learners to the CE monitor for you as well. There's also AMA, APRA Credit 1 categories that can be expected to be received via a PDF certificate via your email. So you will receive all this information if you sign up for this webinar via email, but I'm just providing it to you here so it's in your memory. All right. Final thing before I hand it off to the presenters, we are operating off of Zoom technology, which means you've got the capability to type in and engage with our speakers and type in questions, either a chat box or a comment via the chat box, or a question through the Q&A feature, which is in the bottom, depending where your toolbar is, it's either at the bottom of your screen or at the top. As the speakers continue throughout this webinar, if you're providing questions, we will note them. We will either address them during the webinar or post-webinar. And finally, the webinar is being recorded. This will be available through the CIDRAP website and also through a MAD-ID portal for you to view later. If you have a colleague, you thought this was really great and they want to go watch this and obtain CE, they'll be able to do it there, or feel free to reach out to us if there's any questions you may have. I think that's all I have, so I'm going to stop sharing, and I'm going to invite Dr. Humphries and Dr. Banerjee to join us today. And they are going to start, I think Dr. Humphries is starting off, but they're going to co-present. Dr. Banerjee and Humphries will co-present together. So I'm going to go on mute so you can take it away.
Thanks, Marnie. I put myself on mute so I wouldn't interrupt that, and I forgot to take myself off. So I hope, Dr. Banerjee and I will have an opportunity to share with you all some of our experiences with these new diagnostic innovations that we have available to us and how they can help inform some of our antimicrobial stewardship activities. So next slide, please. So our disclosures are here, and as Marnie mentioned, you can find them online as well. Next slide. And so what we're going to talk about today are some of the novel approaches to microbiology diagnostics, and we're really going to focus in on blood specimens because that's where most of the innovation has happened to date. We're going to evaluate the clinical impact of these technologies and then also describe some post-analytical innovations that you can do, some kind of low tech, some kind of high tech that can improve your stewardship efforts. Next slide. Okay, so when we think about the types of innovation for antimicrobial susceptibility testing or resistance, I think often many of us focus on the analytical portion of these tests. And so we do have new technologies that allow us to get the results more rapidly. We have results that are available that look at genetic content of the bugs, even some genomics, so next generation sequencing innovations, and then certainly even some tests that help us get those MIC values much, much quicker. So there's lots and lots of innovation that's happening here. But the important thing to keep in mind as we implement these new technologies is that we really need to think about the entire diagnostic process. And so the pre-analytical as well as post-analytical aspects of this really need to be considered. And there are things that we can think about and innovate in when it comes to these steps. So, we've talked a lot these days about diagnostic stewardship, so ensuring testing is only sent when indicated. Some of these tests can be kind of pricey. And then from a lab end, we can also make sure that we're not just performing these rapid susceptibility tests on everything, but only really on the relevant organisms. And then on the post-analytical side, those of you who have heard me speak before know I like to talk about breakpoints. So these are the interpretations that we use for our tests. And so if you're using a brand new technology with outdated breakpoints, you're probably not helping patients in the most desired way. So of course, making sure we're using up-to-date interpretations or breakpoints. And then also think about some smart reporting. This could be report comments. This could be cascade reporting. It could be a lot of different things. And the goal here is to partner with your stewardship teams to ensure that whatever the lab is putting out is also helping to support those stewardship initiatives. And then, of course, there are the stewardship interventions that Dr. Banerjee is going to talk a bit about. Next slide. Okay, so like I mentioned, we're going to focus on blood. And so for those of you who don't work in the lab, sometimes I think, we draw the specimens, we tube them off, and then it's like this big mystery to what happens to them. So when we get a blood culture, typically this is collected in a blood culture bottle. We get 2 per set. These are incubated on an automated instrument that's going to monitor for bacterial growth in real time. At some point, the instrument is going to tell us, hey, there's something in this bottle. Then we do a gram stain, we subculture it. The next day, we can do a MALDI identification. Most labs are using MALDI these days. Set up our antibiotic susceptibility tests. And then in another day, we get those results. And so all told, it can take 2 to 4 days, sometimes even longer, depending on the organism, to get through this diagnostic process. And so there are some innovations that can help us speed things up. And so if I go from the right side left, we do have new technologies that are allowing us to perform susceptibility testing more quickly. And so instead of a traditional 16 to 18 hour kind of incubation to do a susceptibility test where we're really waiting to see how the bacteria behaves, there are technologies I'll talk about in this talk that allow us to do this within a couple of hours. That's very exciting. So you could get those results the same shift. The other thing, and that's okay, if we take a look back, there's also directly from when that bottle flags positive, we can perform a rapid identification using a molecular method and susceptibility test. And again, this is from a pre-incubated blood culture. And then if we go even further earlier in the workup, there are some rapid multiplexed PCRs and next generation sequencing technologies that can provide us identifications for bacteria that might be in a blood specimen even without doing a culture. So we're going to kind of talk through this process. Next slide, please. So before we dive into that, I think it's important to kind of understand the different flavors of how we determine antimicrobial resistance in the lab. So the approach that's been sort of tried and true and used, frankly, since Alexander Fleming described it, is the minimum inhibitory concentration test. And so by that test, we're exposing the bacteria to antibiotic at varying concentrations, and we're looking for the lowest concentration that inhibits the growth, that minimum inhibitory concentration, or MIC. The beauty of this approach is theoretically, we don't really care how the bug became resistant. We can detect it, right? It's able to grow in the presence of the antibiotic. That's all we need to know. The downside is we got to give those bugs time to grow and respond and behave in order for us to be able to observe that phenotype. What's much quicker is doing a genotypic test. And so this is when we're looking for a gene, like let's say MAC-A or CTXM, that would confer MRSA or an ESBL. And so we're looking for the presence of the absence of a gene. The problem here is you're only going to find what you look for. And so if you have a new kid come into town that has some new resistance mechanisms, you're not necessarily going to be able to find that. But it can be very quick. And unfortunately, we do struggle a little bit with the no gene detected. What does that mean? And we'll go through a case that describes that. And then finally, and this is a little bit, in the future, because we still don't have a great grasp on it yet, but we're getting there, is just doing a whole genome sequence. And then we can look at all of the genetic features of a given organism and then use those to predict what the MIC might be. And so, like I said, this is largely academic right now, but it's definitely coming. And so something to be aware of. Next slide. Okay, so as I mentioned, I think maybe click one more time. I think there might be a, yeah. So maybe one more. Yeah. Okay. So there is a lot in the space of rapid positive blood culture tests. And we like these because we have a really enriched specimen we're working with, and there's limited bugs in the blood usually. And so we have tests on market today for identification and susceptibility tests. We have tests that give a rapid susceptibility result. There's a variety of them, both specific and CLEX are, well, CLEX is approved by FDA, specific is under review still, I believe. And then we also have tests that give the identification and resistance genes. So different flavors, all kind of with the same goal of providing data much, much quicker than that original timeline that I talked about. Next slide. So like I mentioned, the market is kind of like flooded with these blood tests. And we know that like we're not only interested in blood infections for our patients, but the reason that these are the first sort of samples to get these innovations is kind of multifactorial. But essentially, we all know sepsis is a very clinically important diagnosis. So people are maybe willing to pay a little bit more to get a rapid result from a patient who has bacteremia. There's also a pretty high incidence rate. And so that means if you're a company manufacturing these, there's a good total available market that you can sell these to. So this is not just like looking at, I don't know, cerebral spinal fluid, which also is very important, but maybe less frequent than patients who have bacteremia. And then the samples itself overcomes a lot of the technical hurdles that we deal with when we think about microbiology. So the first thing is like 90% of your blood cultures will be negative, and you don't want to be running an expensive test on a negative sample. And so by the process of doing a blood culture, you've weeded out all of those negatives already. The organisms are also pre-amplified, so you have a lot of signal present in that bottle for your technology to be able to detect. And the reason that's pre-amplified is because the bacteria are growing, they're replicating. And finally, these are usually monomicrobial, and there is a relatively limited number of bacteria that typically cause bacteremia. So the panels that are developed don't have to be huge, all-encompassing. And if they have a hard time with polymicrobial, more than one bug in the blood, usually you can kind of deal with it because it's less frequent. Next slide. Okay, so I mentioned there's a couple of different genetic tests available today. I'm not going to go into the details of these. I think that the trends here to note is, lots of bugs can be detected. If we look at the GenMark panel as an example, 41 different bacterial groups, 14 yeast, 1 mold, 10 resistance mechanisms. This is great. We still aren't quite at the point where we can use these to replace the cultures because while 41 bacteria may sound like a lot, we still see patients who have infections caused by organisms not covered by these. And the other part is we still need that phenotypic MIC to best predict susceptibility. Next slide. Okay, so let's go through a case. And Ritu, I think I'll turn it over to you.
Yeah, great. Thank you, Romney, for that background. So how might genotypic organism ID and resistance gene detection work in a clinical scenario? So let's go through a case. So this is a 30-year-old who suffered 65% total body surface area burn wounds following an explosion. 2 weeks after admission to the burn ICU, this patient decompensates. So at the time of decompensation, the patient is started on vancomycin and cefepime, and we're calling that day 1. The blood culture rapid test is performed, and this is after blood cultures grow and show gram-negative rods on a gram stain. So the ID and the genotypic resistance marker are Klebsiella pneumoniae and Serratia, and a Vim beta-lactamase gene is detected. So that patient, on day 2 that this result is available, is started on ceftazidime, avibactam, and aztreonam, which is recommended therapy for Vim-producing multi-drug resistant gram-negative strains. How is this information valuable? Well, so if we didn't have a genotypic resistance detection marker on this platform, if we just used the gram stain result and looked at our institutional antibiogram, you would say, well, most of our 99% of our gram negatives in the bloodstream are susceptible to cefepime. So you might have kept this patient on cefepime. Within a day after subculture and ID, perhaps through MALDI, you would know that you had Klebsiella and Serratia strains in the blood. And you would still maybe say, well, cefepime is pretty good, or maybe we should escalate therapy to meropenem, which is 99% of our Klebsiella pneumoniae isolates are susceptible to that. But when we had the rapid test, we were able to demonstrate that a Vim gene was detected. And this, of course, is a metallo-beta-lactamase. And so the recommended therapy is not just ceftazidime, avibactam, or imipenem, relebactam. It's actually ceftazidime, avibactam plus aztreonam. So this was getting this patient on appropriate therapy very quickly due to genotypic resistance detection. Okay. But does it always work so well? Well, the answer is no. Careful, you really need to know your epidemiology. So here's another case illustrating that. So this is a 29-year-old with postpartum sepsis. Within 15 hours of hypotension, a blood culture is collected and grows gram-negative rods. The blood culture rapid test shows the presence of E. coli and specifically says that these various resistance markers are not detected, including a CTXM, which is our most common ESBL gene in the US. So based on these results, this patient is transitioned to ceftriaxone. And this is based on information from our antibiogram and our local resistance epidemiology. But unfortunately, 55 hours later, when conventional antibiotic susceptibility test results are available, we can see that the isolate is actually resistant to ceftriaxone. So this patient has been on ineffective therapy for 55 hours from the time the blood culture was collected. Okay, so this is where absence of a resistance gene, as reported in this report of this rapid blood culture test here, does not equate with susceptibility to a certain class of drug. And Romney's going to speak more about this in the next slide.
Yeah, so it is kind of nuanced for gram negatives, right? So we're very used to looking at MAC-A for Staph aureus. That means MRSA. If there's no MAC-A, it means MSSA. It's very straightforward. Gram negatives are all these shades of gray, right? And so while CTXM is the most common ESBL, it is not the only ESBL, and there are over 2,600 different types of beta-lactamases that might be expressed by these organisms, and it's impossible to make a test for 2,600 different targets. And so it does vary, based on where you are. And so you can see across the US, on average, about 20% of ceftriaxone resistance is not due to CTXM. That differs regionally. And so if you look at, for example, the Mid-Atlantic, only 12% is not due to CTXM, whereas if you're in New England, it's 43% not due to CTXM. And if you were able to focus in more narrowly to your individual hospital, what you might see at your hospital could be different than the hospital down the road. And so it's really key to understand the patterns that you see at your own institution as you're making these guidelines. Next slide. So if we focus then on our area, we're in the East South Central region, about a third of ceftriaxone resistance is not due to CTXM. And so if you look, like, what is driving that resistance? Well, there are other types of beta-lactamases, ESBLs, like SHIV variants. There are also a fair number of these E. coli and Klebsiellas that have acquired a plasmid AMP-C gene. And then there's just a whole variety of other types of beta-lactamases that are hard to sort of, delineate in a chart like this. One thing I do want to point to, just because I can't help myself, if you look at the ceftriaxone MICs, the thing that makes it so important that your lab is using current breakpoints is that for isolates that are CTXM negative, they're more likely to have those borderline MICs that we used to consider susceptible, but are now considered resistant by contemporary isolates. And so this is really where, marrying good data from your phenotype and the genotype is important, because if you're using the wrong breakpoints, even with the CTXM test, you're going to be missing this important resistance. Next slide.
Okay, so Romney's really spoken a lot about how many different kinds of rapid blood culture diagnostics are available now. But do we know, is there actually good data to suggest that these rapid blood culture diagnostics actually demonstrate clinical benefit for patients and their outcomes? Well, the answer is there's some data. So most of the studies to date have been small observational pre-post study designs with historical controls, and all of the confounders and all the limitations of those study designs. Moreover, a lot of these studies have demonstrated mixed results, and when you're trying to interpret them all together, what you realize is these studies are very heterogeneous. They look at a variety of pathogens, a variety of resistance mechanisms, and they use a variety of implementation methods when the laboratory first brings these tests online. And a lot of these studies did not actually report out on clinical outcomes. They reported test performance. So really the results have been mixed when you look at the observational data. And what do I mean by that? So this is a list of some of the early observational studies looking at rapid blood culture diagnostics and patient outcomes. It's not an all-inclusive list. And what you can see on the leftmost column is there's a variety of different tests that have been studied. So it goes from FISH to GeneXpert to MALDI-TOF to Banerjee, single gene PCR detection, multiplex PCR tests like the BCID. And then also at the very bottom row, there's a meta-analysis of 31 studies. You can see in the next column over, some of these tests, these studies implemented the test with antibiotic stewardship review, but not all of them. Almost all of these studies demonstrate a decrease in the time to optimal therapy when these tests have been implemented compared to historical controls. Okay, but really, some of them demonstrate a mortality benefit, some of them did not, and many of them did not report mortality. And then some of them demonstrate a reduction in the length of stay. shown as a negative number here. One of the studies showed a reduction in length of stay of 22 days, which raises some eyebrows about what their baseline, routine standard of care is. And then some report cost savings and most do not. So we wanted to know if we could evaluate clinical benefit of a rapid blood culture diagnostics through a more rigorous study design, like a randomized controlled trial. So a few years ago, now several years ago, we conducted one of the first prospective randomized controlled trials, implementing a rapid blood culture diagnostic, which was in this case the BioFire Blood Culture ID Panel, or BCID. This was a single center prospective RCT where in the laboratory, when a blood culture was gram stain positive for gram negative bacilli or gram positive cocci, they were randomized to one of three arms. They would either be in the control arm, which got standard of culture, a standard of care culture and susceptibility testing, which at the time was MALDI-TOF, organism ID, and a PBP2A for rapid detection of MRSA. In the second arm, they got all the standard of care testing, plus they got use of the BCID test. And in the third arm, which was like the Cadillac intervention, they got standard of care testing, plus the BCID test, plus antibiotic stewardship reviews that were conducted 24/7 by an ID physician, who is myself, or a pharmacist. Okay. And this is really the main finding of the study, is that if I orient you here, each row here is a different study arm. You can see control, BCID test alone, or BCID test plus stewardship in the bottom row. And time 0 is when the gram stain was positive. So you can see the organism ID was reported much faster. That's the red box in the two arms that had the rapid test, the BCID test. Antibiotic escalation occurred much faster in the two arms with the rapid test. This is, for example, a patient who had MRSA who was not on effective anti-MRSA therapy. They got started much more quickly on appropriate therapy compared to the control arms. But what I want to point your attention to is this blue inverted triangle. These are the folks that got overtreated, who needed antibiotic de-escalation. And de-escalation happened more quickly only in the arm that had the rapid test plus stewardship review, calling the service and saying, hey, it's okay for you to de-escalate antibiotics based on the rapid test result. So really what we learned from this study is that there's an added benefit of pairing stewardship with rapid diagnostic test implementation, especially for antibiotic de-escalation.
Super. So as I mentioned, we've got the genotypic test, and it has some pros and it's got some cons. And so what we all want is a rapid phenotypic test, right? So this can test all antimicrobials and predict both susceptibility and resistance. And the cool thing about what's going on today is there are so many creative people out there developing all sorts of crazy methods to tackle this problem of having to wait for the bacteria to grow. And so there's a lot of different flavors. Some use time-lapse imaging. And so some of you may be familiar with this. That's essentially what the phenotype blood culture test from Accelerate does. The NGP from Celux also has a similar type of approach. There's a company that has a very fancy cantilever type way to measure or weigh the actual bacteria, with the idea that mass goes down as they're dying. There's tests that look at bioluminescence, which is only made by viable cell. There's cell sorting and counting methods. The reveal system is sort of like a molecular nose. So it's looking for the emission of volatile compounds by the bacteria. If you've ever walked into your microbiology lab, you'll know what I'm talking about. It's a little bit of a odorous place. So you can use sensors to detect those odors. looking at transcriptional changes or even light scattering. And so there's some technologies that are available, FDA cleared today. There's some kind of using my crystal ball that I think will be available in the next 1 to 2 years. And then of course, what's not shown in here is that rapid disk diffusion method that both CLSI and UCAST developed, which is pretty low tech, but is very effective as well. So really, all labs can consider implementing some of these rapid direct from blood susceptibility tests. Next slide. So it sounds like, time-lapse imaging, that's pretty straightforward. We'll just look at the bacteria. I do want to say it takes a lot to be able to predict what a bacteria is going to do down the road. And so this is just an example for Accelerate, where you can take a look at the complexity of data that goes into these predictions. And so they're looking at things like cell morphology. Is the bug filamenting like we see with some beta-lactams? Does it become very circular depending on the PBPs targeted? It looks at the cell morphology, surface area, the division morphology, replication kinetics and dynamics, et cetera, et cetera. So there's a lot of data points that go into this. And so you compare all of those features to a growth control, as well as databases and prediction models to get that end MIC. So it is pretty sophisticated what people are coming up with. And that level of sophistication is needed because bacteria, they behave in all sorts of exciting ways, and there's always outliers. So you've got to be able to be ready for those. Next slide. So this is a study that we did when I was at Accelerate, and I think it just kind of highlights one of the challenges if you have one of these rapid phenotypic tests. And so we took a look at patients who had been enrolled in our registry study. So this was multiple hospitals across the US that had implemented the test. And we looked at before they had the system and after they had the system. And we stratified patients based on their pit bacteremia score, which is kind of like an academic measure of how severely ill the patient is. We don't use this clinically, but it is a helpful tool from a research perspective. And so what you can see is that for patients who had mild pit bacteremia scores, meaning like they probably did not actually have a true sepsis, it was probably just a blood culture contaminant, there was really no difference of having the novel technology. When you looked at people who were moderately ill, we were able to show a substantial reduction in the time it took when you use the technology to get patients on optimal therapy. However, if you kind of bumped up to patients with severe bacteremia or severe pit bacteremia scores, you lost that benefit. And I think this kind of speaks to something with human nature. We like to see the patient stabilize a little bit before we start making these big changes to their treatment regimens. And so that is probably what's feeding this. And then the other thing you have to keep in mind is these patients who are critically ill have a lot of heterogeneity in the group. So they may have co-infections, they may have higher risk for multi-drug resistance, all of these types of factors, which again make it more difficult to optimize therapy very quickly with these results that are available soon. Next slide.
Okay. So again, we wanted to study clinical impact through a rigorous study design of a randomized controlled trial. And here we used the Accelerate Pheno test, which Romney just went over. And we did a 2-center prospective randomized controlled trial where we just enrolled patients with gram-negative bacilli in their blood cultures. And we randomized to one of 2 arms. Both arms in this trial had baseline antibiotic stewardship review of the rapid blood culture test. And the stewardship team would call the treating teams to make recommendations, either to escalate or de-escalate antibiotics if needed. And the control arm had standard subculture and AST methods. The intervention arm had the standard culture and AST methods, as well as the Accelerate Pheno test. And here's what we found. So what I'm showing you here is a graph of the percent of patients who had a change on the left, on the y-axis, and on the x-axis is the time from randomization from when a gram stain was positive on that blood culture bottle through 72 hours that we followed each patient. And the control arm, the standard of care method, is shown in red, and the rapid method, the accelerate pheno group, is shown in blue. So you can see overall antibiotic changes happened about 6 hours faster in the rapid testing arm. That's not that amazing. But then when you look specifically at time to changes in gram-negative antibiotics, we're starting to see more difference between the treatment groups. So the rapid testing group had antibiotic changes made a median of roughly 25 hours faster. Okay, so that's interesting. But what was really interesting to us was how the antibiotic changes varied by resistance of the bloodstream isolates. So when you look at just those who had resistant organisms defined as ESBLs or carbapenem resistant gram negatives in the blood, what I'm showing you here, again, the control arms are red, the rapid testing arm is blue. The solid lines have resistant isolates and the dashed lines have susceptible isolates. So you can see there's a significantly faster time to change in antibiotics for those with resistant isolates in the rapid testing arm compared to those with resistant isolates in the control arm. Okay, almost 2 days faster. And this is antibiotic escalation. So presumably these are patients who had resistant organisms that were not recognized and were not on effective therapy. So the escalations happen significantly faster when there was rapid testing, okay? What happens if you have susceptible organisms? So in that case, it's the antibiotic change is not really antibiotic escalation, but it's rather de-escalation. And susceptible isolates are shown with the dashed lines. So you can see de-escalation happened faster in the rapid testing group compared to the control group, but the difference was less dramatic. It was 13 hours faster. Unfortunately, we did not see any significant differences between the treatment groups in clinical outcomes like mortality. And we can talk about why that might be later on in the question and answer period, but it might be that we had relatively small numbers of patients with resistant organisms, and mortality is a very complicated outcome that may have a lot more to do with comorbidities and severity of illness of the patient than how quickly we get them on the right antibiotic. Okay, so just to illustrate this a little bit more, we'll go through a case. So this is a 14-year-old neutropenic patient who develops fever, vomiting, and hypotension, and is admitted to the ICU. But prior to admission to the ICU, they are started on cefepime in the ED. The blood culture rapid test identifies E. coli, and the rapid phenotypic AST results demonstrate resistance to cefepime, but susceptibility to meropenem and piptazos. So very quickly, this patient is escalated from ineffective therapy to effective therapy on day 1. So presumably, this patient then goes on to have a good outcome, which is a terrific use of a rapid phenotypic AST. But does it always work so well? And as Romney alluded to, not always, especially when you're dealing with very critically ill patients. So this, again, is a 14-year-old neutropenic patient who develops fever, vomiting, and hypotension and rapidly decompensates in the ED and is admitted to the ICU. This patient is started on meropenem in the ED. They were so worried about her, they went for the big gun. The blood culture shows Pseudomonas aeruginosa. The rapid phenotypic results demonstrate this isolate is susceptible to cefepime and meropenem and piperacillin and tazobactam. But this patient is continued on meropenem until she stabilizes, until her fever's gone and her hemodynamics are improved. And this is despite the availability of the rapid test results on day 2. So this, again, as Romney mentioned, means that providers are reluctant to de-escalate antibiotics and do less if patients are extremely ill and until they've stabilized.
All right, so that's kind of what we have today, which is all very exciting, but you know, what is next? And wouldn't it be wonderful if we lived in a world where you could collect a specimen and then get your ID and AST results really quickly and at the point of need? And I want to say to you all, I do think there's possibilities for this, and I think next generation sequencing might be what gets us there. Now, there's always been some limitations to next generation sequencing. Traditionally, it's been very expensive. That cost has come down dramatically. It's been very labor intensive to do the library preps, get the samples ready. Now automation is available. And the time to results has always been a challenge. It usually takes like 72-ish hours traditionally to get these tests done, but the time to results are decreasing all the time. And so we're getting there, but we do still 100% rely on that phenotype, which a next generation sequence test does not provide. Next slide. So there are a lot of folks out there working on these challenges. I think many of you may be familiar with the T2 system, which is an identification system. There's others like Qvela, Momentum, Helix, 3IDX that are working on direct from blood identification of pathogens in the blood. T2 has a limited menu of 5 organisms, but they're critical organisms, right? So it's either Candida in one test, or it's, you know, the bugs we worry about antibiotic resistance for, like Staph aureus, Pseudomonas aeruginosa, that type of thing. There's targeted next generation sequencing. So DNAE is a company that is working on this challenge. And so they have very broad panels, or there's broad range next generation sequencing. And again, many of you may be familiar or have heard of Carius test, which is something that is a reference lab test that's performed off plasma, and it looks for cell-free DNA. But there's other companies like Day Zero, PathoQuest, and others that are working on this. I will say, remember what I said at the beginning of the talk that like 90% of your blood cultures are negative? Well, same thing is true for these tests, right? They may detect 1 or 2, you know, handfuls of more cases, but the majority of samples that are sent to yield a negative result. And so there are, you know, obviously limitations to this, and these technologies today are pretty expensive. Next slide. So I mentioned that we don't have a great way to predict antimicrobial susceptibility from a genome. And so there's really been sort of 2 general approaches to this problem. So one is what we call like a lookup table. So it has a long list of all the resistance genes and then what antimicrobials that particular gene might impact, right? So it's like just a lookup table, what's going on. This is challenging because bacteria often use resistance mechanisms in concert, and so you don't get the full picture if you're just kind of doing a one-to-one tabulation. The other approach is machine learning. And so machine learning is when you look at all of the features in the genome, you don't really care what they are, and you compare those to your desired phenotype, the MIC that the organism has, and develop some artificial intelligence type algorithms to predict the MIC based on all of those features. Now, there are a lot of challenges before this comes to fruition. Some of the challenges today is while we have lots of whole genome sequences and databases, few of these include MIC data. Furthermore, those that do, the MIC data are often not reference methods, they're commercial methods because that's what everybody uses, but there are limitations to those commercial methods. And so you don't have like a good reference result. The databases usually only include 1 or 2 antimicrobials as class representatives, but we know that that's tricky, right? Like meropenem and imipenem might have different resistance pathways. And usually these databases only have information on susceptible, intermediate, or resistant, not the actual MIC. And that's a problem because, as we all know, those breakpoints or interpretations are changing all the time. And then finally, if you're testing a mixture of organisms, it's really hard to say this bug has this gene and that bug has that gene. You get like a mixture and it's hard to define who belongs to who, which does become important when you're thinking about mixtures of organisms like Pseudomonas aeruginosa and E. coli. Next slide. So here's some just data of the lookup table approach. This is using Patrick and NDARO. And so these are big databases, and they evaluated it against two different lookup table type approaches. One is called CARD and the other is ResFinder. And so they did find major errors. So that means false resistance ranging from 25% up to 42%. And so you can see in the figure, CARD is in the light gray and ResFinder is in the dark gray. And so these are balanced accuracies for a variety of organisms. And so, just to kind of give some examples of the types of limitations, if you're looking at efflux genes, so these are genes that pump the antimicrobials out of the cell, that only has about a 52% positive predictive value for antimicrobial resistance. Aminoglycoside modifying enzymes, same thing. Beta-lactamases are a little bit better, and fluoroquinolone resistance genes and mutations are even better yet. So you can see by drug class, things vary a little bit. Next slide. So as I mentioned, machine learning is an approach where you look at all the features in the genes, you correlate that to MIC data. You don't necessarily need to know the resistance mechanism. And actually a cool thing about it is you can discover resistance mechanisms using this type of approach. And so the performance, though, is dependent on how good the database is. And if you have a good mix of susceptible and resistant strains that represent global isolates, because we know antimicrobial resistance travels with our patients, the relative complexity of the resistance mechanisms, the plasticity or how quickly an individual species mutates, and then sampling bias, like I mentioned. So geographical or time frame or demographics can all introduce noise. Getting to an MIC is really tough with this technology today, but getting to a susceptible or resistant looks like it is something that's feasible in the near term. So next slide. Just to give an example, we took a look at one of these databases and we just looked at E. coli and we just looked at cefepime. And what we found was actually excellent prediction of one of these machine learning databases from a company called NGD, where we had 95% agreement of the reference method, so broth microdilution with broth microdilution, so the reference method with itself, kind of highlighting that none of our tests are perfect, but 97% agreement of the machine learning approach with the modal MIC. And that included correct calls of susceptible for ESBLs that were missing the resistance factor that causes the bug to turn on that ESBL. So a lookup table would say these are resistant when in reality they're susceptible. So that was really good. Next slide. Ritu.
Yes, so this is an illustrative case of how a next generation sequencing test might actually be helpful. As Romney mentioned, these tests are often negative or they provide results that are difficult to interpret, but this is a case where it worked well for us. So this is a 3-year-old female who had very refractory juvenile idiopathic arthritis. She had been treated with multiple immunosuppressive medications and biologic agents. She was extremely, extremely immunosuppressed. And she continued to have pancytopenia that was getting worse and worse. So she was admitted and had multiple blood cultures that were negative. We weren't convinced that she actually had an infection at all. We thought maybe this is her underlying disease. But we did send a next generation sequencing test from the blood to a reference laboratory. This was the Carius test. Within a few days, we got this result back, which showed lots of copies of Mycobacterium avium complex in the blood. They also detected lower copy number of Klebsiella pneumoniae and HHV7. This prompted us to send a dedicated mycobacterial blood culture on this patient. We weren't quite sure the significance of the Carius test result, but we wanted to confirm it with standard culture. And it took a little bit of time, but by day 13, a week after we collected the blood culture resulted with positive stain for AFB. And then by day 22, the conventional culture had identified Mycobacterium avium complex. So this is an example of how the next generation sequencing test more quickly identified the causative agent in the blood than a culture. But as many of you know, it doesn't always work so well. And these are very complicated tests to interpret. So this is an example of that scenario. So this is a 52-year-old female with a very complex genitourinary history. She had repeated history of UTIs, and she was very poorly managed, multiple courses of antibiotics, and concern for drug-resistant organisms. So a next generation sequencing test was submitted to a reference laboratory, and the report, the actual report that was returned back is shown here. There were high copies of Proteus, medium copies of Enterococcus E. coli, Klebsiella, and Candida, and then low copies of various other bacterial organisms. And then there were also resistance genes that were detected, and the report tells you what antibiotic class this resistance gene conferred resistance to, okay? And then the comments from the report were additionally confusing. So it said, well, there's an SH-SHIV combo. These genes are associated with resistance to all of these classes. But in patients with cystitis caused by fluconazole-resistant Candida, they can be treated with amphotericin B. So there's treatment recommendations here. They're also recommending Cipro plus fluconazole for first-line therapy, and then also some alternative therapy. So very complicated to understand what this means, and it's not clear that any of these are actually the causative pathogen in this case. So what some institutions are doing are actually creating committees of experts to actually help interpret these complicated reports like this, and actually provide some recommendations that are easier to follow for treating providers. So as Romney mentioned, there are lots of tests out there. What do the data show about the clinical utility of these next generation sequencing tests or the universal broad range PCR tests that some of us use for very challenging clinical scenarios where we think there might be an infection, we just can't find the organism through conventional testing. So here are just a couple of studies. Most of them are cohort studies, either prospective or retrospective. And they are looking at various tests, and some of them have a substantial number of patients. Like the first one here has 200 patients. The second to last one here has 800 patients. But several of them are also smaller, single centers with 80 patients or so. And the important thing to note is the positivity rate where they actually detect organisms varies, okay? But it's generally low with the next generation sequencing result only. So on the order of 6%, 3%, 4% here, okay? And this is only when the sequencing result provided a result that the conventional testing did not, okay? And then if you look at the very last column, that shows you what are the other outcomes. Did the sequencing result actually lead to a change in management? And most of these studies are saying probably not. So for example, 7 of these 13 cases who had a next generation sequencing result that was positive would have affected treatment, but that's 3% overall of all the patients enrolled. In this study here, the Hogan study, there was a negative result or no clinical impact in 93% of patients, okay? So the summary of the data to date are that the yield of a lot of these tests is very low. There are few outcome studies, and it's really unclear at this point if it's worth it. And if it is worth it, for what patients and what specimen types should we use these tests? And the jury is still out on a lot of these questions. So I want to conclude here with just a few words about implementation of rapid testing. And this is relevant not just to rapid blood culture tests, which we focused on in today's webinar, but all kinds of newer molecular rapid diagnostics for infectious diseases that are increasingly available to us as clinicians. And the important thing is to realize that there are many steps in the process from offering a test to actually getting optimal care to a patient. So there are pre-analytical and analytical steps, obviously, that the laboratory performs to get us a rapid test result. And then there needs to be communication of some form to the provider so that they can get the result in a timely way, of course, otherwise, why are you doing a rapid test? And that they can understand and interpret the result. And then they need to act on the result in order to modify their management and get optimal treatment to patients. So it's a little bit like a parachute. It is only useful if you know how to use it, right? Or you know how to implement it. So it's really important to think about implementation plans. This is in collaboration with your clinical microbiologist, of course. And what kinds of things can we do to nudge clinicians to act promptly on rapid test results so that they can modify their antimicrobials appropriately? Well, there are a lot of things that many institutions do post-analytical, on the post-analytical side. So one of those things is cascade AST reporting. So if you have an isolate that's susceptible to narrower spectrum agents, the laboratory reports those out, but keeps the susceptibilities to broader spectrum agents hidden unless clinicians call and ask for those results to be reported, okay? There's audit and feedback by the stewardship teams, and this often does not happen in real time. It might happen, say, the day after a result report is available. And it's not feasible necessarily to do this on all positive results. So it really involves some strategizing with your stewardship team about what results should they be performing audit and feedback on, what kinds of tests, what kinds of specimen types. The microlabs are excellent at providing comments within their result reports, sometimes linking to treatment algorithms, infection control recommendations, recommendations to consult the infectious disease team if they're highly resistant isolates, for example, and also references to the institutional antibiogram. Some electronic medical records have decision support systems available embedded within the EMR. And then there's also real-time ASP interventions. So, for example, if you get a critical result in the microlab, maybe you page the ASP pager so that they're aware of a critical result so they can help the treating provider interpret the result and act on them. So communication of results is really important. You probably cannot read all this text, but this is a lot of language that has one critical result, which is that this is a Staph aureus detected and a methicillin resistance gene, MEC-A, detected. But there's a whole lot of language here that you're asking busy clinicians to sift through to understand what it is that they need to act on, right? So maybe a simple thing like making this critical result that's in a box in a different colored font, if that's possible, or bolding it would be helpful. For the BCID trial, the first trial I talked about today, we put some treatment guidance in the result comments, and we vetted these with our infectious disease clinicians. We thought they were clear and straightforward, but then when I showed them to non-infectious disease clinicians, they were confused. So, for example, the second row here, which says Staph aureus MEC-A not detected, preferred therapy is an anti-staphylococcal beta-lactam antibiotic. The response I got from some of these non-infectious disease clinicians was, just tell me what to use. What is an anti-staph beta-lactam antibiotic, right? So we really need to think about how can we communicate more clearly so we can nudge providers to do what the appropriate therapy is in this case, right? Other implementation issues to consider. So not only how was the test communicated to providers, but how can the test be integrated into the lab and the current clinical workflows and stewardship workflows and algorithms that you have in place? And this is going to vary from institution to institution, from lab to lab, right? what is the cost effectiveness of these tests? Now, these, for the most part, are not replacement tests. These are add-on tests to current standard of care testing. And so they're always going to increase cost. But what is the number of tests you need to do in order to have optimal impact for a few patients? Okay, and this is an open question, but it's really worth thinking about. And then what about operational issues? What happens on nights and weekends when the laboratory has less staffing and when you probably don't have stewardship available by pager? And what happens in community hospitals where the number of cultures that you're working up might be lower and where the staffing is not equivalent to larger tertiary quaternary care hospitals? Okay. And then I think it's also important to highlight to clinicians the challenges in interpreting novel blood culture diagnostics. We talked about this already, but the significance of nucleic acid detection is not always clear, right? It might not reflect a pathogen. It might reflect a colonizer, especially, I don't know what low-level detection of nucleic acid means in every clinical scenario, right? And how can we help clinicians interpret results like this? And then there will be discordance. If you're doing both genotypic and standard of care phenotypic AST, there might be discordance, right? And so what is the testing algorithm that the lab uses to evaluate this discordance? And how do you report that? And what do you tell your treating providers? And then always remember the limitations of molecular diagnostics, and perhaps your stewardship team should be the ones that are liaising with the treating teams about this. The absence of resistance genes does not always equal susceptibility to a given drug class. And a lot of these PCR-based diagnostics might have poor performance with polymicrobial cultures and not necessarily pick up all the cultures that, all the organisms that are in a given culture. Okay, so I'd just like to end here and say that rapid blood culture diagnostics are increasingly available, very exciting, and have great potential to improve time to optimal therapy and patient care. And these tests not only give you more rapid organism identification, but also now susceptibility testing and the potential for direct from specimens identification. But remember that few studies actually demonstrate improved clinical outcomes using a lot of these rapid diagnostics. They definitely do demonstrate shortening time to optimal therapy, and that's maximized when implemented together with antibiotic stewardship reviews. But there's a real need for more implementation research to understand how to optimally integrate these kinds of rapid tests into clinical practice. So with that, I just want to thank you very much for your attention. And Romney and I are available now to answer any questions you might have.
Excellent. This is superb. I see we've got some participants coming on a little bit later in the webinar. Wanted to remind everyone that this was recorded. And so you'll be able to re-watch this if you missed parts of it through the CIDRAP website. And obviously don't forget to collect your CE. So we've got some questions that have come in. Romney, I don't know if you've had the chance to look at any of those, or Dr. Banerjee. And then I have a pressing one, but I'll hold. I'll let you, if there's one that's burning there, I'm going to let you, if you want to pick which one you'd like to.
Well, so I think there was one question about whether or not the curious test can really be considered rapid if it takes 40 hours to come back. And I think it kind of depends on the situation, right? I think that the data is emerging that it's really fabulous for mycobacterial and fungal type infections where we know our standard of care can take like 20 days, 30 days, 40 days. So compared to that, it's rapid, but compared to something a little quicker, no. And I think, as Ritu mentioned, lots of hospitals are developing committees on kind of like what are the indications of use for this type of testing. And one thing we always try to stress is we don't want to send this off, until at least we've had our routine standard of care cultures have had a chance to incubate at least a day or two, right? Because often those will be available and can provide answers much more quickly and significantly less expensive.
I have my question is just something that seemed like a theme that stuck out is the 90% negative blood culture.
Oh yeah.
I'm trying to figure out how to ask this question. To me, it's from the rapid diagnostics, is it because we're not culturing because we're going direct from blood? Is it a limit of detection? But it also, because a lot of times blood is a surrogate for infections that are in other sites of the body. Because we were focused on the blood culture and rapid diagnostics right now, but there's more localized type of sampling you can do as well. Does that change the percent positive the more you get? So I don't really know how to ask it because I think it's multifactorial, but I want your opinion on this 90% because that may stick out in people's mind listening.
I think it's because every patient who comes in through the emergency department gets a blood culture collected.
Okay.
I mean, I'm being a little facetious, right? But yeah, I mean, we order and send blood cultures on everyone. And there are certainly some, routine practices like for pneumonia, I get a blood culture. I mean, the yield of a blood culture for a pneumonia patient is like next to zero. And so I don't know that we are all that great at blood culture stewardship. There's great work coming out of Johns Hopkins on this, if people are interested, but I think that's really what it relates to. And then of course, you've got patients who are pre-treated and the bugs won't grow in that instance. And then there's also things that, it's kind of the luck of the draw, right? A lot of patients aren't continuously bacteremic. And so if you happen to draw the blood culture when they don't have, bacteremia at that site, you're going to miss it. But keys to improve your yield are, testing the right patients, getting enough blood, those types of things, getting it before you start antibiotics, basics.
So you're not dealing with a negative answer quite a bit. And then I guess how, as an antimicrobial steward, are you dealing with the fact that this is not a false negative? I don't know, Dr. Banerjee, if you want to answer. I mean, you've got some beautiful case scenarios, but I'm thinking how many times you get called in a day with some of these results from, a rapid test, and how am I supposed to interpret this?
Yeah, I think it's the blood culture interpretation is no different. Well, we often have positive blood cultures that we're also interpreting whether or not it's a true pathogen or a colonizer, right? And that really depends on the clinical scenario. So it would not be any different if you get an organism detected through, a rapid genotypic method or phenotypic method. You always treat the patient based on the clinical scenario, not necessarily a laboratory result.
Yeah.
Right.
I mean, I think one of the overarching objectives, I mean, we have several objectives we were addressing with this webinar, but one of the bigger conversations that we were having is, here's this technology that's either recently FDA approved or, as Romney presented to us, it's coming in the next 24 months or so. And how are we as antimicrobial stewards going to be knowledgeable, vetting this within your hospital systems? I think your implementation, at the back end of this webinar was really important. And just, understanding how to interpret for the best treatment of the patients. And I think that's a big goal. And I think it's continuous education and learning, because it's getting more complicated, potentially. That's a little bit of my soapbox, I guess.
But I do think it's important to think about, like, the next best fast, exciting test might not be the next best test to implement for impact on clinical care. Like, it's exciting, but we really need to study in what patient populations would these tests have the optimal impact. I think there's a question here asking if we would, if it's more value in allocating rapid diagnostics for high-risk patients than for all patients, all comers, all specimen types. And I think that's a very interesting and important question. One that we're thinking about all the time, right? Because all of these tests add additional cost. Maybe we need to really hone down on the patient population in which they're likely to have optimal impact.
Yeah, and it's not always the patient in the ICU, as we demonstrated, right? They may be more reluctant if you we've often talked about, like, let's just focus on urosepsis, because these patients, you can get them out of the hospital pretty quickly if you can optimize therapy pretty quickly, and that may have a big impact, to the overall healthcare system.
Romney, I'm just posting the
Yeah.
Dr. Humphries and Dr. Banerjee. I'm just posting the continuing education QR code there for people to be able to go receive their credits, or they can reach out to the MAD ID organization. We've just got some questions how to go about receiving the credits. So there was a question around how you felt this is going to be utilized, rapid diagnostics, whether it's microorganism identification or, moving closer to antibiotic susceptibility and phenotype in an outpatient setting. Where do you see that utility happening now or in the future?
I mean, I'll give my take on it, and I'd love to hear Ritu's as well. I think that there are definitely roles for some of these technologies, and we've seen it, right, with COVID, having COVID tests, rapid tests available that are very accurate in the community setting is great. Rapid flu tests, rapid group A strep tests, all those things that we have now that, don't need a culture to back up, or don't need a test to be sent to a lab to back it up. It is tricky from a reimbursement perspective. So some of these technologies that you might consider using for a patient who's outpatient, like a gastrointestinal panel for a patient with diarrhea, or a respiratory panel for a patient, with a cold, those aren't being reimbursed today if they're broad panels. And the idea is that the evidence just is not there to support the use of them. So I do think we have to be cautious in how we implement these things, because we don't want to wind up having, these mandates like we will never reimburse for this. And if you give an example of what's happened with the respiratory viral testing, it has huge value for some outpatients, patients who are severely immunocompromised, value for some pediatric patients. But there was kind of this knee-jerk response from the insurers that they were not going to cover any at first. And that took a lot of work to kind of overturn. So, I think to Ritu's point, we need to study these things so that we can provide the best evidence possible. And then that's how we can implement them.
I have a question, I'm trying to look at the questions here, but I'm wondering what's so terrific about having the two of you here today is you not only are striving to understand the technology and its best utility, but then you study the outcomes when it's under practice. Where's your research focused right now? Because to me, those are the papers that are going to come out in a 12 to 20, 12-month period. So I'm just kind of looking into your questions that you're having, which might help us gauge where the science is moving.
Well, we are.
That way you can share with us.
Yeah. So we are doing a trial looking at one of the direct from blood technologies, looking at a stewardship endpoint for that. So that'll be a pragmatic randomized controlled trial. So that'll be exciting to see, I think. I am always working with companies on the development of these new diagnostics, kind of very early all the way through. And you probably picked up on it as I was talking about it, but the next generation sequencing early work is really interesting to me. And so we're working a lot on how can you marry that from an epidemiology perspective, a patient care perspective, all of these pieces, because there's potential for all of that. So that's kind of what I've been working on. Ritu?
Yeah, and I've been working on a multinational trial. It's not enrolling yet, but hopefully soon, in areas of the world with much higher gram-negative resistance rates than we have in the US, to try to see, because that's the population where we think early detection of resistance is likely to have the most impact on getting patients on appropriate therapy quickly, and then maybe leading to a downstream mortality benefit. So we're working on trying to do a similar outcome studies to the blood culture trials I presented, but in areas with much higher resistance rates, because we think that's the population that would benefit the most.
Yeah. Final question, because I apologize for those of you that came on basically the time zone and came on an hour late, but we're only, this isn't 1 hour, 15-minute webinar, so we'll be ending here soon. But please watch the recording at a later time if you missed any parts. The question I want to take from one of our participants is around, seems like the industry technology developers are interested in the outcome, and now there's the opportunity to engage with other organizations or even regulatory bodies, and you're both involved in different organizations and giving speeches and things like this. So the question is just, it seems like there should be more involvement in advancing the implementation of this type of research to better understand which organizations do you feel are involved or need to be involved.
Yeah, I will say, you know, Ritu has worked very extensively with the ARLG. That's how those trials got done. And, you know, I think kind of the mandate of the ARLG is to do these studies that might not otherwise get done, but are still very clinically important. And so we've had success working with the manufacturers for investigator-initiated grants to get some of this work done. And then there are also some funding mechanisms of like a small business kind of partnership type grant, like a cyber grant, so they're called, from NIH. So that's all possible. And I think, you know, for all of us who do this work, it's important to think, you know, we're great at doing analytical studies, like how well does this test work? But at the end of the day, if it's not making an impact on patient care, who cares how well the test works, right?
Yeah.
And so I think, you know, it's kind of on us to start pushing that science forward and looking at more than just the analytical, you know, piece of it, but also the clinical impact.
Dr. Banerjee, do you have anything to add?
So the ARLG, which Romney mentioned, is the Antibacterial Resistance Leadership Group that is part of NIAID. And they're always looking for concept proposals. So if people on this webinar have ideas for implementation studies to look at clinical impact of diagnostics, I think they would be interested in seeing that.
Excellent. Well, I'm going to have to close because we are out of time. I want to thank Dr. Humphries and Dr. Banerjee for putting these slides together, all of their extensive work that helps others implement programs, and really just all your insights and your time. Just mentioning again, for people to reach out to us, there'll be a follow-up email with more details for how to view the webinar again that's been recorded and collect your CE. And that's it. We'll close and thank our sponsor, our support from the educational grant, for being able to bring these speakers to you and these insights, and that is from bioMérieux. So thank you to our sponsors supporting that. All right. Thank you all. Have a good day.
Thank you.